What makes the human brain so special? Have you ever wondered? Imagine this. There are many intelligent species around us: chimpanzees, crows, octopuses, dolphins, and although it might seem funny to us now, any one of these could potentially become intelligent enough to build a civilization similar to ours. We can imagine a world where, instead of a species with our bodily form, there are beings resembling other primates like those in the Planet of the Apes movies, or more octopus-like creatures that could build civilizations and traverse intergalactic distances, as wonderfully depicted in the movie Arrival. What I'm trying to say is that having a body like ours is not a prerequisite for being intelligent. For example, we can imagine a planet where superorganisms, much simpler in structure than us like ants, but working with extraordinary cooperation, could achieve a level of intelligence comparable to ours either individually or collectively, and build cities, vehicles, and technologies similar to ours. But we are not living on such a planet, and as far as we can see right now, it doesn't seem like such a planet exists. And when we look at our only example, this pale blue dot, we see an undisputed leader in intelligence. We humans. And when we ask what has given us this leadership, there is undeniably only one organ that stands out: our brain. Therefore, there must be something special that sets this organ apart from those of others. But what? I mean, what distinguishes this piece of flesh inside our skulls from what's inside the heads of other animals? And more importantly, is there a possibility that other creatures could one day build civilizations like ours? Let's take a look. If you recall, I previously detailed the 10 reasons behind the evolution of the human brain to its current size, shape, and function. Therefore, in this video, I won't delve into the evolutionary history details of how we became so intelligent. If you haven't watched that video, I highly recommend you do so. Today, we'll focus more on the point we're at right now. In other words, we'll take a narrow slice of the 4 billion-year evolutionary history and compare a few living organisms within that slice to uncover what makes the human brain special. If you want, let's start by opening up the brain and looking at its general shape and diagram. Then, we'll gradually delve deeper. When we look at the brains of different animal species, the most noticeable difference between theirs and ours is the size difference. It seems that, when it comes to brains, the bigger they are, the better. Think about it this way. As Homo sapiens, our brain is around 1,500,000 centimeters. The brain of Homo erectus, our ancestors who lived from 1.9 million to 140,000 years ago and are also known as erect humans, was about 950 centimeters. For the Australopithecus africanus species, which are common ancestors of the approximately 14 different species under the genus Homo, and lived from about 3.3 to 2.1 million years ago, this number was only around 450 centimeters. Do you notice the pattern? The further back we go in our evolutionary history, the smaller the brains of humans and human-like creatures become. Our living close cousins are not much different either. A gorilla's brain is about 550 centimeters, and a chimpanzee's is around 400 centimeters. But these last two numbers might surprise you. Because a chimpanzee is much smarter than a gorilla, yet a gorilla has a much larger brain. Indeed, similarly, an elephant's brain is much larger than ours, and a whale's brain is even larger. Therefore, mere absolute size is not sufficient. Because we know that larger-bodied animals also have larger brains, just like their hearts, kidneys, and other organs. Thus, we need to look at the ratio of brain mass to body mass. This ratio is called the encephalization quotient, or EQ for short. But this EQ is not the same as the emotional quotient, EQ. Don't confuse them. If an animal's EQ is equal to 1, it means the brain size is exactly as expected for its body size. If it's greater than 1, the brain is much larger relative to its body. If it's smaller, the brain is smaller than expected. According to this coefficient, humans have an IQ between 7.4 and 7.8. That is, our brains are about 7.5 times larger than expected for our body size. An African elephant, which has a much larger brain than us, has an IQ of only about 1.3. So, the IQ of elephants is just 1.3 compared to ours. This explains why elephants, despite having large brains, cannot build civilizations. By the way, if you're curious, chimpanzees have an IQ between 2.2 and 2.5, macaques around 2.1, and mice around 0.5. As you can see in the evolutionary process, our brains have become far larger than they should be. This is truly an extraordinary level of awareness, but there's more. Because when we look more closely at our excessively large brains, we see that not every part of our brain has grown proportionally. Some brain regions have grown much more than others. The cerebral cortex, which handles many advanced human qualities like planning, reasoning, and language, is the outermost layer of the brain and has grown significantly more. Also, the cerebellum, located at the back of our brains and fed by a large number of neurons, which coordinates our movements, especially hand-eye coordination, and thus our ability to make tools and plan, has also grown more than expected. This disproportionate growth is one of the main reasons why our higher cognitive functions are so advanced, as I previously explained. But when we strip away the overall anatomy of the brain and look deeper into the neurons, the nerve cells that make up that general anatomy, we also see certain differences. The most notable of these is the number of neurons. For example, the human brain has 86 billion neurons, which is 1,000 times more than a mouse's neurons and 13.5 times more than a macaque's. Or, to put it another way, a mouse's brain contains 71 million neurons, a macaque's 6.4 billion, and a human's 86 billion. This isn't actually that surprising. After all, our brain is as large as those of these creatures, but these neuron counts show that the enlargement of the brain isn't just an empty balloon. As the number of neurons increases, the complexity of the neural network processing data and generating responses also increases, leading to greater diversity and richness in the types and variety of reactions to stimuli, just like we see in artificial intelligence research. But we also need to recognize a fact. For a creature to be intelligent, it doesn't necessarily need to have a brain like ours. Very different biological architectures can create similar results. For example, think of crows, some of the smartest animals on Earth. Despite having only about 2.2 billion neurons, they can be trained to perform complex tasks and remember them for long periods, even though crows' IQs are similar to those of tailless monkeys, around 2.2. And of course, since birds and mammals share common ancestors, their brains have many similar structures. But overall, the brain anatomy of birds is very different from that of tailless monkeys. Yet, they can be so intelligent. Therefore, we need to look for a deeper factor that causes intelligence. Researchers seeking to find this factor are examining the differences between neurons. Actually, doing this is more difficult than looking at the brain as a whole or its parts. It's even more challenging than simply counting the total number of neurons in a brain. Because you would need to examine each neuron individually, determine which category it belongs to, and catalog it. This is an extremely tedious and costly task. For example, the entire brain of a mouse hasn't been fully cataloged for its 86 billion neurons. But thousands of scientists conducting research in this direction continue to work diligently. Thanks to these studies, a total of 300 different cell types have been discovered in 100 different regions of the human brain so far. But this number is expected to increase significantly in the coming years, because even in mouse brains, where all brain cells have been cataloged, a total of 5,300 different cell types have been discovered. Interestingly, these different cell types don't seem to provide much information about why the human brain is so advanced. Of course, there are certain regions where specific cell types specialize. For example, in our visual cortex, which processes visual information, some neurons appear to exist only within that structure. But researchers have not yet found any neuron type that can be said to be unique to humans. The same neurons seem to exist in other animals, albeit in different forms. Not in terms of cell types, but when looking at how a single cell type appears in two different creatures, we encounter some morphological differences. For example, a group of cells called pyramidal cells, named because of their pyramid-like shape, are much more common in the human brain compared to a mouse's brain, and these cells become much more triangular and branched, living up to their names. In mice, however, they are much stouter, shorter, and have fewer connections. In chimpanzee brains, we see a shape that falls somewhere in between, just as we would expect in the evolutionary process. These pyramidal cells have a functional meaning as well. The upper parts of the pyramid connect with the higher layers of the brain, while the lower parts connect with the lower layers. Therefore, when a pyramid is longer and more complex, communication between different brain layers becomes tighter and more intense. For example, the part of the pyramidal cells in the human brain that connects to the cortical layer is much thicker than in chimpanzees and mice. This might allow the neural activity deep within our brains to be processed more intensively in the upper layers. Let's delve even deeper. Neurons are ultimately just cells, aren't they? Therefore, like other cells, they are produced and controlled by genes, and genes, in turn, interact with the environment to determine what types of neurons progenitor, or precursor, cells become during the embryonic and childhood stages. Thus, as these genes change through the evolutionary process, the structure, frequency, and connections between the neurons that make up the brain also change. For example, when we look at the auditory cortex, which processes sound information in both mouse and human brains, we see that in mice. A type of neuron called excitatory neurons is much more prevalent compared to inhibitory neurons, whereas in humans, the opposite is true. As their names suggest, excitatory neurons facilitate the spread of signals, while inhibitory neurons regulate neural activity and suppress it. Additionally, in the human auditory cortex, the proportion of non-neuronal supportive nerve cells, such as astrocytes, oligodendrocytes, and microglia, is about 5 times higher. These cells support the activity of neurons and allow the connections between neurons to strengthen or weaken as needed during development. This disproportionate growth does, as I mentioned earlier, explain the main reasons behind our advanced cognitive functions. But it's difficult to know exactly what these differences mean for the superiority of human intelligence. However, as the number and depth of brain atlases cataloging these differences increase, and with the assistance of rapidly developing artificial intelligence, it's very likely we'll discover patterns we couldn't see before. Thus, we'll be able to understand the human brain much better. Now, let's also look at connections. Because, as you can imagine, no neuron operates in isolation. There are always other neurons it connects with. Which neurons they connect with, how they connect, and when they fire determine whether a neuron will fire in any given situation, thus directly determining whether a particular process related to that firing will occur. For example, in a study examining 2,000 neurons from the cortices of mouse, macaque, and human brains and the 1.6 million connections between them, it was shown that the connection map, or connectome, of the human brain contains 2.5 times more interneurons than those of mice, and these interneurons make 10 times more connections with each other. Interneurons regulate neural activity and control firing. They are intermediary neurons. Therefore, the number and frequency of their connections directly affect how the brain processes stimuli and the level at which it does so. Additionally, bipolar interneurons, neurons that connect with each other but rarely with other types of neurons, are very rare in mice but make up more than half of human interneurons. In contrast, multipolar interneurons, those that enjoy connecting with other types of neurons, seem to be less common in humans. For example, this last finding might explain an interesting paradox we observe in humans. You know that neurons work extremely fast. Imagine this. The fastest neurons in the human body can transmit signals at 120 meters per second. Even if you were only 2 meters long, this shows that signals could be transmitted from one end of your body to the other in just 16 milliseconds, which is 20 times faster than the minimum time your consciousness can perceive. Moreover, the distances within our brains are much, much shorter than this. Despite this, the formation of our thoughts and even the shaping of the movements we exhibit generally takes at least a few seconds. This might be explained by the fact that the inner neuron network in the human brain is more complex and larger than that of other species, potentially slowing down the completion of neuronal activities and the formation of more complex thoughts and movements. Finally, let's look at one more critical difference that ties all of this together: the speed of development. As I explained in the video about why human babies are so helpless, the human brain develops much more slowly compared to other animals. While a deer, an elephant, or a giraffe calf can start walking and running shortly after birth, it takes human babies months before they can even perform meaningful actions. Don't ask me how I know this. Researchers observe that no matter which part of the human brain they examine, they see a much slower developmental process compared to other animals. Think about it. Reaching the adult brain size in a mouse is completed within about 5% of the mouse's average lifespan. In macaques and chimpanzees, full development is achieved in about 33% of their lifespan. In humans, it takes about 30 years, nearly 50% of our modern lifespan, especially considering the lifespan expectations of our ancestors. In this case, speed isn't actually an advantage, especially when it comes to producing a brain capable of building civilizations. This slow development rate allows the precursor cells I mentioned to remain in their undifferentiated states for longer periods. As a result, a much larger number and greater diversity of neurons can form in the human brain. These neurons, thanks to the extended time, can achieve much greater diversity and sophistication. Additionally, this long developmental process allows the neurons in the human brain to interact with the environment for longer periods, providing more opportunities for shaping. This facilitates our ability to adapt so effectively to our surroundings. Here's an interesting detail. The precursor cells I just mentioned can differentiate into more diverse cell types. For example, the progenitor cells in rodents can only differentiate into one type of neuron. Therefore, there is little reason for their slower development. In fact, we have even managed to solve the mechanism behind this slow development. A specific gene duplication in the human genome, where a gene is accidentally copied to have two copies instead of one, seems to be the main cause. When we duplicate this gene in mice, their neurons develop more slowly, and their learning abilities increase. Perhaps in the future, thanks to what we've learned from these brain studies, we'll be able to produce large brains like ours. Thus, we won't have to wait for the slow and tumultuous processes of evolution. Who knows? That's it. All that I've explained is a brief summary of the remarkable differences we've discovered between human and other animals' brains through anatomical, general structure, histological, tissue level, cytological, cell level, and genetic examinations. As you can see, aside from relative size and some minor structural differences, we don't see significant differences between human brains and those of other animals. Depending on how you look at it, this might be surprising or not surprising, because the fact that such large differences in cognitive abilities can arise from such small differences shows us the power of biology. All right, another point. Different environmental conditions, genetic pools, and randomly distributed circumstances could allow a completely different animal species to become a dominant brain species and build civilizations surpassing humans. But at the same time, the smallness of these differences is not that surprising, because this is exactly what we expect from the evolutionary process. We humans did not descend from the sky. We evolved from other animals. Therefore, we should expect certain specializations and minor variations. And we do find them, but these differences should be negligible compared to our commonalities. And indeed they are. Of course, there is still much we need to solve, but as we find more pieces of the puzzle and, with the help of rapidly advancing artificial intelligence, discover patterns we couldn't see before, a more consistent and solid story emerges. And this story is the most magnificent detective story illuminated by humanity. Mine, the story itself is the brain. Truly, the only organ that can discover itself. See you in the next video. Goodbye.